Today we're going to be talking about chemical sensory transduction, taste and smell. Taste and smell are chemical sense. They give us information about the chemical composition of our surroundings. Let's start our discussion about the chemical sense of olfaction. Our nose can detect and separate many different types of compounds. The map of these compounds occurs by matching our information of memory templates stored in the brain. Some of the basic attributes that the sense of olfaction are: what's the sense of smell, where it's coming from, how strong is it, and what's the timing of the smell. When we talk about the sense of olfaction, we talk about two major organs. One is the main olfactory organ, which is called that encompasses the main olfactory epithelium, and in many animals the vomeronasal organ, also known as the accessory olfactory epithelium. As you can see in this diagram, the main olfactory neurons projected the main olfactory bulb that then projected to many different brain areas, while the vomeronasal organ projects specifically to the accessory olfactory bulb that then projects to the ventral amygdala. Now in this diagram is shown the major cell types that's found in the main olfactory epithelium. As you can see in this diagram, the olfactory sensory neurons have this very polarized type of structure, with cilia in the apical end and a cell body and a thin distal end that send the axons to second order neurons. Now, in close proximity to these olfactory sensory neurons are the supporting cells. The supporting cells provide, as the name implies, metabolic support and also structural support to the main olfactory sensory neurons. These olfactory sensory neurons are replaced constantly every few days, so the replacement of these neurons derives from these basal cells, which is a type of stem cells that you find in this kind of epithelium. These axons from the olfactory sensory neurons terminate in these second order neurons that are contained in structures we call glomerulus. The structural polarity of this olfactory receptor neurons is also reflected by a functional polarity. So if one records electrical responses of this olfactory receptor neurons to very specific odorants, one finds that the odorants are able to elicit big responses when applied to the cilia of these olfactory receptor neurons, but much smaller responses when applied either to cell soma or to the distal end of these cells. So the molecules that are able to bind to the odorants and perform the transduction process are concentrated here in the cilia of these olfactory receptor neurons. There is much to be learned about how these specific molecules that we call odorants are able to be recognized by these olfactory receptor neurons, but now we know that these molecules are G protein coupled receptors, and specific receptors, of specific olfactory receptor neurons respond to multiple odorants. So, for example, recording from this cell shows that these cells respond best to this specific odorant, while this another cell can respond to two different types of odorants, while a third cell can respond to those three types of odorants. So odorants can activate many receptors, and receptors can respond to multiple odorants. The identity of these receptors were uncovered by using genetic techniques, and the surprising fact was that these odorant receptors, which are expressed in these olfactory receptor neurons, are the most diverse type of G protein coupled receptors found in the body. Actually, in mice, about 1,000 different genes encode these receptors. These receptors are, are very diverse, and we think that each neuron, each olfactory receptor neuron, express only a single type of receptor. In humans, we have about 10 million olfactory receptor neurons. Approximately each neuron has about 1,000 receptor proteins. So we think there's about 1,000 different types of olfactory receptor neurons in humans. And we can discriminate at least 10,000 different odors. And interesting fact is that, for example, dogs that can be much more sensitive to odors than humans, about 10,000 times more sensitive to odors than humans. Each olfactory receptor neuron has the same sensitivity in the dog compared to a human. So what makes dogs so much more sensitive to odors is one fact is that there is many more olfactory receptor neurons in the dogs, which is about 1 billion receptor olfactory receptor neurons in the dogs than just 10 million in humans. Now let's discuss the sensory transduction pathway that's involved in olfaction. Everything starts with the bind of the odorant to the olfactory receptor, which is a G protein coupled receptor. This olfactory receptor will activate G proteins called G-OLF, which is similar to a GS type of G protein. This activation of G protein will activate adenylate cyclase type 3 in the olfactory receptor neurons, which will break down ATP to form cyclin P. Cyclin P then can bind to cyclonucleotide gate channels, and the bind of the cyclin P to these channels will open this cation selective pore in these channels and will allow the influx of both sodium and calcium. An interesting fact here is that these cells also express chloride channels that's calcium sensitive. So when calcium enters through these channels, calcium will activate these chloride channels, which will allow the chloride to leave these cells. The sum of these activities of these receptors and channels is a depolarization of these cells that will further release neurotransmitters to second order neurons. These olfactory receptor neurons also show the phenomena of adaptation as any other kind of sensory cell. So if an odorant is applied for a long period of time, here over 5 seconds or so, you can see that the response of this cell wanes very quickly with time. Another way to see this phenomenon of adaptation is to apply odorants at different time points. So odorant applied at time 0 here show this very strong response, and the same odorant applied uh, 1 second or so later display a much smaller response because of this phenomenon adaptation. Now if the odorant is applied 2 seconds later, the response recovers. 4 seconds later recovers even more, and 5 seconds later the response is almost back to 100%. So this phenomenon of olfactory adaptation is something that you can relate in your everyday life as very commonly we can sense a smell, for example, inside our house very quickly, and then with time we sense that smell much less, and sometimes we don't even perceive that sense that particular smell, even if the uh, if the smell is still there. Uh, and one of the reasons why we have this adaptation is that, again, like in the photo sensor system, that amplifies the dynamic range of these cells. So, for example, here is application of uh, of a odorant that elicits a very specific response in the cells, with increased response with larger uh, durations of odorants. Uh, if the olfactory receptor neuron adapts, then there is this shift to smaller responses that if you look for the overall curve, basically there is this increase of the dynamic range of these cells to the odorants. Now there are many aspects of olfactory receptor neuron adaptation that kind of remind us about phototransduction or photoreceptor adaptation. Here is an interesting experiment in which a whole cell pipette is used to record the odorant responses. So application of odor here elicit this response in this olfactory receptor neuron. Now if the same recording is made in the presence of BAPTA inside of the pipette, now you can see that this wane of the response or decline of the response with this odor is much diminished. So explain the effects of BAPTA odorant cell responses. Speculate some of the molecular mechanisms that underlie the effect of calcium in olfactory transduction adaptation. And finally, compare the effects of calcium in olfactory and receptor adaptation. Another interesting electrophysiological experiment is using chloride channel blockers. In this case, a cell has been voltage clamped, or olfactory receptor neuron has been voltage clamped, odorant has been applied for a very brief period of time, and here is the response elicited by this odorant. Now the same cell in the presence of this chloride channel blocker in the outside elicits a much smaller response. So So explain the results here in terms of olfactory transduction cascade. So what's the direction of the chloride currents in this case? So what would be the function of these chloride channels in the olfactory transduction cascade? And finally, what's the expected chloride clear potential in these cells and why that would be the case for these cells? We're going to be talking about this particular signaling cascade, but basically the signaling cascade, this diagram depicts the major elements involved both in transduction and also adaptation in these olfactory receptor cells. And notice here that not only this diagram now shows a little bit more detail the presence of this molecule called calmodulin. So when calcium enters the cells via the cyclonucleotide gate channels, calcium can bind to calmodulin and calcium calmodulin can bind cyclonucleotide gate channels. Moreover, this calcium can affect these chloride channels and also affect a multitude of enzymes here in these cells. Also, finally, as shown here, some of the other components that are involved in recovery of the signaling cascade. So other molecules are beta-arrestin, pKa, and also IGS-2.